less emphasis on molecular epidemiology. And then maybe at some point when there is uh, some space or some slot, practice session on the quote unquote aligner can be carried out later.
Right, right. So, Malvika, thank you so much for recording in advance. You can start the recording, please. So friends, thank you so much, for the penultimate day. I take great honor and pleasure in having Pushpendra Singh, who is scientist E2 at NIRTH, National Institute of Rural Tribal Health in Jabalpur. He's a group leader in Mycobacterium leprae group. Prior to that, Pushpendra was at uh, uh, Dr. Hans Laboratory in Baton Rouge in US, where he was a visiting scientist and also he served as a senior postdoctoral fellow. And then he returned back to India in 2016 as Ramalinga Swamy Fellow, where he joined as an assistant professor, and then he joined this ICMR Institute. He obtained his PhD with the very famous Dr. V.M. Katoch, who was an ex-ICMR director. And he obtained his PhD from National Jalma Institute in Agra, which is again an ICMR institute, where Pushpendra worked on comparative genomics of pyrazinamide, pyrazinamide resistance in mycobacteria. And then he also served briefly, not briefly, in fact, for almost 6 years, where he served in EPFL, which is a very famous institute in Lausanne in Switzerland. He was there for almost 6 years, where he worked on comparative transcriptomics of leprosy. His group has been steadfastly working on leprosy, and they have done some groundbreaking research in the areas of mycobacterium leprosy, which is I would say understudied in many areas, and they have developed wonderful methods, approaches, and Pushpendra and his group have published in a myriad of papers, including Nature and Science, which is a kind of an amazing entity that his group has been working for. Last year, Pushpendra received a Bioclues Innovation Research and Development Award, and that was when we requested him to take up uh, uh, as a vice president or chair for Bioclues, and he has been honing this wonderful position as vice president research for Bioclues since last year, and he has been associated as a core member. So, with this little introduction, I kindly request Pushpendra to deliver his talk. So, as he said, there will be a quick 1 hour talk with about 10 to 15 minutes demo. Following which, if you have any questions, from other exercises, we'll be doing that. Okay. Yeah. Thank you so much, Pushpendra, in advance for your time. Yeah.
Thank you very much. I would start sharing slides. I hope the slides are in slideshow mode.
Yeah. Yes, please. Yeah.
Okay. So, good evening, everyone. Thank you for joining for today's session. It's my pleasure to be part of this wonderful series, and I'm very happy to see this response actually from all of you, this regularity and the insistence from Prashant about maintaining this, because it's actually a discipline. Nothing comes very easy in life actually, and if things are priority, then we need to spend time out for those. And I really appreciate you taking time for continuity of this series, and especially for the entire team of Bioclues who basically give so much time for organizing these things so regularly. Thank you, Prash, and it's actually a wonderful feeling to be part of this series. So, without wasting much time, I'm basically going to introduce about molecular epidemiology with particular emphasis on leprosy studies that we have carried out in last nearly a decade, and it basically represents work carried out in around 38 countries, uh, and some of this was during my postdoctoral work at EPFL, that's Swiss equivalent of the Indian IITs, for example. So it's École Polytechnique Fédérale de Lausanne, which is a French word for Swiss Federal Institute of Technology, similar to Indian Institute of Technologies in India. So, as Prashant mentioned, that I have mainly worked in microbacterial genomics and drug resistance during my PhD, and subsequent to that, one of the interaction during this PhD actually opened a way for postdoctoral research in Professor Stuart Cole's lab. This has been a wonderful uh, experience of my life. That is why I say that some of the conference, not every conference may change your career, but some of the conferences will very certainly have this potential of changing or giving you a very good direction. So yes, please make good use of your conference experiences. So because that really was very helpful for me, because I was working on TB drug resistance mechanisms, and since the institute was Leprosy Research Institute in Jalma in Agra, I used to take active participation in all those conferences also. And during one of these conference, got a chance to ask some questions from Professor Stuart Cole, and he gave me exactly the same question to investigate it for the next 6 years during my postdoctoral work on leprosy. And from there onwards, I have never left the field of leprosy because it's so interesting. And I will try to show you how this actually, why it seems to be so interesting to me. And besides that, we would also get to know that leprosy is not a disease that has been eradicated. It's important to understand this. Uh, so, for my academic journey, basically, after my next day after my PhD, I was fortunate to get into Professor Stuart Cole's lab in Switzerland, in Lausanne. It's a very small city near Lake Geneva, and from there, I got some collaborative work with National Hansen's Disease Program, which Hansen's disease is actually another name for leprosy in Baton Rouge in USA, and we proposed a collaborative study there. For 2 years, I worked. For 6 years, I worked in EPFL, and then for 2 years as a visiting scientist in Baton Rouge. And then, what project I carried out in Baton Rouge, I wrote another with the help of other partners. I applied for project which led to my Ramalinga Semi Fellowship, uh, and with that I joined at MS University of Baroda in 2016, and in 2019 I joined ICMR, and tomorrow I will be completing 4 years of my joining ICMR, and that is why all of our lab members are having a get together tomorrow, and that is the reason that I am going to keep this talk slightly brief, because I have to prepare for their party tomorrow. I hope you would excuse me for that. But this is all their work that I am going to present, and this is the teamwork, because, as I mentioned, it's a study in nearly 38 countries involving more than 1,200 samples. Uh, so the genomics of leprosy basically revolves more about the molecular epidemiology and phylogeography. It's actually not a culturable organism. We will know more details in next slides. And there is well-known fact that certain animals can have leprosy naturally also. We would also know about that in next slides. And we know that leprosy does not exist in Europe currently for last 3-4 centuries actually, but it was not the case earlier, some 10 centuries ago. So we would also get to know what was the molecular epidemiology of leprosy in medieval Europe or some 1,000 years back, and how this field is so interesting actually to trace back the history of not only pathogen evolution but also the human migration. And uh, we have had a wonderful talk by Professor Gyaneshwar uh, recently on population genomics, where he mentioned about human evolution and their genomic associations. Um, Similar work, because with human migrations, human pathogens also migrate along with humans. So some part of that can actually be traced back by studying pathogen genomics also. And since pathogens are actually smaller genomes, so sometimes it becomes easier also to study pathogens. So let's come to leprosy first. As I mentioned, leprosy is not a disease of past. Nearly quarter of a million new cases are recorded globally. This is a fact from last 1 decade. So this has not changed much. Only during the COVID pandemic, this underreporting has led to some decline, but other than that, nearly 200,000 cases, 2 lakh new cases are recorded globally, of which 60% are from India. And if the cases are detected later on or with some delay, this can lead to permanent and or irreversible disabilities. Most of these disabilities are very apparent, such as this type of sinking nose, where the oropharyngeal cavity has certain damages from due to bacterial growth, and these type of uh, damages in hands. I would just take the pointer. There's a pointer. So you can see these type of damages on the peripheral parts of the body, because this bacteria likes low body temperature. The optimal temperature for growth of this bacteria is around 33 degrees Celsius. And why it is so, that is also very interesting, because this is hidden, those clues are hidden in the genomics. It's heat shock uh, heat shock proteins are absent or have become pseudogenes. So this bacteria likes low body temperature, and all those clues actually come from genomic insights, and those genomics based inputs actually also guide us for the molecular epidemiology. So just for some idea about what is molecular epidemiology. So basically, epidemiology is the 3Ds actually, distribution. Uh, so determinants of disease basically, those are the those could be demographic determinants, those could be social determinants, uh, pathological or some pathogen based determinants. So all those are part of epidemiology. But if we study that by the molecular tools targeting the DNA and RNA, then it's mainly the pathogen genomics based molecular epidemiology. And we are today talking about with the special example of leprosy, because this is not a culturable organism, so we have to depend on genomics for understanding its biology and as well as its transmission. So coming to the transmission, most of you would be surprised that we do not fully understand how leprosy is transmitted from one person to another. The most commonly believed mechanism is that it transmits from one person to another through droplet infection, just like any other airborne disease is transmitted. But leprosy has this distinction of being one of the slow growing, slowest growing microorganism, to which nearly 95% of the human population is resistant to develop disease. It's very reassuring that despite infection or despite exposure, less than 5% of the people will develop disease, because it's there could be some host genetic factors also. But another factor is that this bacteria has learned to be in harmony with its host. It doesn't want to damage its host until very late, when its number outgrows and it starts to manifest the clinical symptoms. That takes nearly 5 to 7 years on an average, and some people may not never develop disease actually despite exposure. And the longest incubation period for this disease has been 32 years documented, because one of the chimpanzee which was imported at the age of 2 years from West Africa, it developed disease at the age of 32 years in Japan, and it showed the same strain that is present in West Africa. So that confirmed that the incubation period was of more than 30 years. So all this is very interesting, and how this actually proved, it's because of the molecular epidemiology. By determining what is the type of strain present in this chimpanzee, uh, and what are what is the geographic distribution of those strains, whether those strains are present in Japan or not. If those are not present, where those are present, and is there some epidemiological link between those strains uh, present in the human population in West Africa, from where this chimpanzee was imported at the age of 2 years only. And in zoo animals, they are given regular checkup. So it was beyond doubt proven that this animal manifested disease after 30 years, and molecular epidemiology helped in proving that. Otherwise, it was very important to know the origin of leprosy in this animal, because in Japan there are almost no cases of leprosy these days. Earlier there were very high number of cases, some 40 50 years back, when there was no chemotherapy available for leprosy, or no effective chemotherapy was available around 6 to 7 decades back. At that time there were a lot of cases of leprosy in Japan also. So people were very keen to determine the origin of this infection, whether it is from Japan. If it is the case, then you need to focus your attention on checking or stopping the transmission. Um, so that is the value of molecular epidemiology, and with that it also proved that it can take so long also for developing disease, and there are complex host genetic factor for susceptibility to this disease. But let's see one interesting story where we can go back before the time of Christ, or the before the Common Era BCE. Traditionally, it is known that leprosy, the first very accurate description of leprosy is in from India. In Sushrut Samhita, it is which is 600 years before the Common Era, or we also call it as before Christ. So nearly 2,600 years back, there are some ancient texts named as Sushrut Samhita, where it is very clearly mentioned by Sushrut that what are the clinical symptoms of leprosy. It even mentions the exact cardinal signs which are even today considered very accurate and characteristic of leprosy only. For example, the loss of sensation, the discoloration of the skin, and uh, these type of characteristic lesions. It also mentions about the cholmura oil, which has some efficacy against leprosy, and that was actually used for centuries until the modern chemotherapy became available. Since our ancestors knew so much about this disease and they had described it, so it was believed that leprosy was present in India, and from India it went to other parts of the world. And that was a kind of stigma on entire country, that India is responsible for the spread of this disease. It's actually unfortunate to label a place with a disease, because common people only blame uh, common people would not actually appreciate the details of pathogen evolution. It doesn't take place overnight. So linking one place with the origin of any pathogen is actually not very scientific, because these pathogens have actually occupied this planet before us. We are only 100,000 years old. Modern anatomically modern humans are only 100,000 years old when they were in Africa, and then from with their migration it went to other parts of the world, and that has been shown by several genetic studies, by mitochondrial DNA based studies, by Y chromosome based studies, as well as language based studies, because language also evolved with the human migrations. But can pathogens also help us in retracing the human evolution and migration, or the peopling of the planet? So this study was carried out by Professor Stuart Cole and his team, one very talented technician in Professor Stuart Cole's lab, Dr. Mark Mono. Currently, he's a PhD, but when he published this, he was only a technician pursuing his PhD, and he has so many publications in Nature Genetics, Science, and in so many reputed journals where they publish this study. So you can see that um, traditionally it was believed that Indian strains went with Alexander's army around 300 before Christ, 300 years before the Christ, uh, and they went to Europe. And there has been actually so many documents with this evidence that Alexander's army took back leprosy bacilli with them and it went to Europe. But when Professor Stuart Cole's team investigated around 400 samples from around the world, from all the continents, they found that the strains present in India are of type A, type 1. We will see the nomenclature, but we can say that this is a distinct type. It's known as type 1. Whereas the strains present in ancient bones from Europe, because currently there is no leprosy, but in from the skeleton samples of around 1000 years old samples from graves, it was found that they had some characteristic lesions of leprosy. We will see some pictures in next slides. Those are of type 3. So this is not consistent with the previous theory that leprosy went from India to Europe. When I uh, went there in Switzerland in 2008, this publication was already uh, published in 2005 in journal Science. So the first introduction that Professor Stuart Cole gave to me was that our study takes away this stigma from India that India is responsible for spreading leprosy to the rest of the world. In fact, they are very advanced from so early time that they could very accurately describe the disease and its prevention and the treatment. So he always considered India in very high esteem for its traditional knowledge and diversity. And I was very happy to work in his lab to to see and hear these words several thousand kilometers away from your home is actually very motivating, and I want to share this with you. And talking with the evidence is further substantiating all this, that leprosy actually evolved somewhere in Middle East or in this part of China, and along with the Silk Road it went to Europe, and this is consistent because the strains present in China are also type 3, and those emerged into types of type 3, and from there onwards, with the colonization, actually the leprosy went to North and South America, and those strains are still present in South American countries such as Brazil, Venezuela. Colombia, like that. And the other type 1 strain actually reached to India very long back, uh, maybe with the human migration from Africa to India. And from type 2, they emerged into type 1. And all these are actually consistent with the other evidence of human migration. And one more type from the Africa went to West, from East Africa to West Africa, it evolved into type 4. And with the slave trade, this type 4 reached to South America, where the same strains of type 4 are still present in Brazil, in Venezuela, and several South American countries. And you will see that in all the islands which were actually on the route of these sea journeys at that time have a mixture of multiple genotypes. For example, here you would have so many different genetic diversity. There is type 4 in green color, there is type 3 in pink color, and then even the type 1 strains are also present in uh, these islands. For example, these Caribbean islands, or um, i- here also in these islands, where which were important part of stoppage of the ships for several months actually, when they would basically get enough food, and they, some of those would still remain there. And those were actually the melt pots of different strains. So you still see a lot of genetic diversity in islands along the sea routes. And this is also very much from the simple genotyping scheme. Uh, so we now know that it is the Silk Road that contributed in transmission of leprosy to Europe, and from by colonization it reached to US and South American countries. And somewhere armadillos also got infected with the same strains of which were introduced by European colonization of America. However, lot of this, so all this is based on very limited understanding of pathogen genome, because not every part of the word is represented by the and whole genome of M. leprae or Mycobacterium leprae, which causes leprosy. You would also get to know that Mycobacterium leprae is not the only causative agent of leprosy. There is one more species that we described uh, as its full genome in 2015. It was described by Professor Han from America in a Mexican patient. So we would also know about that evolutionary aspect of leprosy and its causative agents. So you will see here that there has been tremendous success in control of leprosy. You will see that prevalence has come down drastically. But after 2005 onwards, there has been a stagnancy. The new case detection rate has kind of plateaued for India as well as for Brazil. So yes, we do not deny that there has been no progress in leprosy control, but the new case detection rate has not actually declined substantially in last uh, 1 decade. That is why it's important to study leprosy, why those are not actually further declining. And 200,000 cases per year is not a small number, especially when a significant number of those cases actually develop physical disabilities, which leads to their lifelong dependence on others. So let's see this typing scheme, which was developed by uh, studying the genomes of Mycobacterium leprae, how they, what it tells about the genotic leprosy in areas where armadillos, these cat-sized animals are present. In pictures, you may not notice that this how big is this animal. It may look like a mouse to you, but let's see the real size uh, of these animals. This American kid, is so happy to hold this as a pet, and you would see that this is the real size of this animal. It's not a small animal actually, and these animals do not breed in captivity. So it's very challenging to study these animals for any disease or to use them any as a model for anything. But people love to eat them as uh, exotic delicacy in southern United States and in many South American countries also. And
Really, they eat this?
yes, as a very like a delicacy.
Oh my god.
And this is very common as a roadkill also because this animal has very low vision actually. When it is fast approaching car comes against it, it uses its traditional way of protecting against other animals, which is to straight jump up, which is very intimidating for other animals. What this animal is certainly doing, but against a fast approaching car, this doesn't actually work because it directly hits the car, and very often you would see these animals as a roadkill, which and these animals are called as leprosy bag. These are full of leprosy bacilli when they are infected because they have low body temperature, they have very low resistance against leprosy bacilli, and you can see that this is actually full of leprosy bacilli. When somebody is eating this improperly cooked meat, they are actually exposing them or experimentally inoculating themselves unknowingly.
So, what is the common name of this animal?
It's called nine-banded armadillo. Dasypus novum cinctus is the scientific name, but the common name is armadillo. Uh, in Hindi, it's
Ah,
actually
armadillo. Oh, yes,
there is
yes.
no Hindi name that I am very accurate.
Whether we are having this animal in
Uh,
India, I
no,
don't
no,
think.
this is not present in India. Yeah, it's not present outside of the Americas. But people used to love these souvenirs of these animals for several years. And there were farms actually which were capturing these animals. It's actually not a protected animal because it grows in large density. So people hunt them also and make these souvenirs. And at that time, it was realized that some of these animals are naturally infected with leprosy. Uh, and it was not known whether there is any role of these animals in transmitting leprosy to humans. So and it's also very well known from the skeletal evidences that until 1600 year until 16th century, the leprosy skeleton from America are considered sorry, the skeleton samples of humans or other animals, for example armadillo skeletons, are also not having any evidence of leprosy until 16th century, before the uh, European colonization, before the Columbus discovered America. So somehow the leprosy actually reached America, and these animals actually biologically amplified it, and now they are giving it back to humans if you directly contact them or but and this was known from 1970s. People used to blame leprosy researchers that they accidentally released leprosy through some patient dressing material in wild, and these animals acquired leprosy bacteria because of leprosy research activities in Baton Rouge in USA. So to disprove that, what my second postdoc supervisor, Professor Richard Truman, he did was that in his freezer he had samples from 1960s where these animals were not used for leprosy research, and in those serum samples he could prove that even in 1960s these animals were leprosy infected by showing the evidence of antibodies against leprosy bacterium. So these type of uh, old samples are actually sometimes very precious to prove or disprove our hypothesis. And this actually was another relief from the molecular epidemiology viewpoint and from the serological viewpoint to have this type of evidence that it is not to be blamed on the leprosy researchers. First, it was that it is not to be blamed on India for their scientific heritage, and then second was that it is not because of the leprosy researchers' negligence. So yeah, let's see what scientific evidence actually proved that this is there is some link between human and armadillo leprosy. So for that, there was this study carried out to sequence the entire genome of leprosy strains from different parts of the world. And when we see this, let's say, imagine them as some grapes, and on this grapevine, you have suddenly a pumpkin hanging in this phylogenetic tree, a simple phylogenetic tree. You see that a single small circle is representing only one case, but all of a sudden you have this big size of clustering. 28 of these whole genomes are from armadillos, and 25 of these samples are from, in fact, humans. And a large number of these 25 human samples have some direct or indirect exposure to armadillos, and they basically share exactly identical genome, nearly indistinguishable by at nearly at the genome level actually. So along this grapevine stem, uh, you would see these numbers 6, 1, 2, 3, 29. So all these 53 samples have no differentiating marker. They are exactly identical, meaning that they have acquired infection from a common source in some recent years, in last maybe 10 years, 20 years. Because since then they have not had time to diversify. And there are some animals which have accumulated just one snip here, one snip here, two here, one, two, like that. So this is a very strong evidence that these animals are and the human samples have the same source of leprosy. It doesn't still prove that whether the armadillos gave leprosy to human or human gave leprosy to armadillos. But that is not actually the point also. And the reason for that is because leprosy incubation is so long. So in that background, now I hope that you would understand why I spent so much time on explaining the background of this chimpanzee which exhibited its disease 30 years later on, despite regular clinical examinations. So here you would understand that these have same source, and many of these human samples have history of exposure in the areas where these infected armadillos are also present. So this was a very strong evidence and was considered as a possible zoonotic leprosy in southern United States. Professor Richard Truman, he has been working on this since 1970s, but this molecular epidemiological evidence that basically proved and convinced the reviewers that this is actually probably a zoonotic leprosy. When we were writing this paper, we didn't use the word probable, but one of the reviewer insisted that because of such a long incubation period, probably it would be more scientific to use this word probable. And when reviewer says something at from such a journal, we accepted that their comment, and we use this probable zoonotic leprosy in southern United States. Anyway, in subsequent studies, we further proved that this is also spreading to other parts of the United States in southern parts where these animals are present. And in those studies, in published in emerging infectious disease, we didn't had to use the word probable because message was clear by that time. So this is one of the most favorite example of molecular epidemiology. Only we are not saying that this publication came out in New England Journal of Medicine. In fact, when this paper accepted, I was not even aware of the impact factor of this journal. And when somebody told me that uh, if this publication was so important and so nice, why didn't you submit it to Nature or Science? and I had no answer. I said that it's medical journal, more doctors read it, and in fact that was the main reason that we submitted it to New England Journal of Medicine. And just within the embargo period itself, we were actually receiving so many emails, and on WhatsApp, Facebook, and social media. This was very widely publicized because people started telling that we would we were not aware that these animals actually carry leprosy. We would not eat these animals anymore. And if we accidentally come across some road kill armadillo, we will wash our hands. We will take precaution washing of our car and such things. So that is the real impact of any research. It's not in numbers of impact factor, but in how many people's behavior you were able to switch to a desirable behavior with a scientific evidence. So that is my favorite example of leprosy. Molecular epidemiology changing people's behavior for their own safety and for the better effective control of a disease. Another example is about the history of leprosy in Europe. We were talking about that leprosy is no longer present in Europe currently, but it was widespread in Europe until 12th to 13th century. In fact, there were more than 20,000 leprosy hospitals in Europe until 10th century. After 14th century, it started to decline. Uh, in this picture, you can see that leprosy people were forced to take a bell so that others can identify that somebody is approaching them and he has leprosy. So people would stay away from them. They were having separate places to stay. In fact, in some places they were considered as living deads. So all their property can be inherited legally by the next offspring or next member of their family if a person is diagnosed with leprosy, because they basically were not considered part of society. They had to live in leprosarium separately until 14th century when the plague actually spread in Europe. So the church actually used to support these leprosy hospitals, or basically those were isolation centers. But during the time of plague, these were actually very ignored people. So most people might have just died of hunger during this 14th century time of plague, uh, especially when they were uh, leprosy patients. So probably that led to some changes, but we are not sure why it actually declined. Whether the strains which were present in 14th until 14th century had started to lose their virulence, or there was some immunity acquired by the most of the population, and this has some significance. If we say that leprosy eradicated from Europe in 12th to 13th century onwards, then because those strains were not virulent, then this has a different implication for the leprosy control in other parts of the world currently. But if those strains were virulent and still this disease can be eradicated practically without any drug and vaccine in 14th century Europe, then that has a different message to the rest of the world which struggles with leprosy currently. That means that there is some other uh, socio-economic component or host genetic component. So that is why we were keen to study the molecular epidemiology of leprosy in medieval Europe, which is the 10th century Europe. So roughly 1,000 years back. So how to study that? So as I was mentioning that, so the key question that we had was that what led to the sudden decline of leprosy in 14th century? Was it because of the host genetic factor? Was it because of the socio-economic factors? Because half of the European population actually declined during 14th century due to plague. Did it change the socio-economic factors of the post-medieval Europe, or was it because of the loss of virulence of M. leprae strains? So, with our set of samples and skill set, we investigated this third possibility of loss of whether it was because of the loss of virulence of the medieval M. leprae strains. So, for that, we collaborated with several um, archaeologists which had access of leprosy skeletons from different time points of Europe, and these skeletons had characteristic osteological evidence of leprosy. For example, this is tooth would have this type of shrinking here. The nasal spine is damaged here in this otherwise well preserved skeleton sample. This type of porosity of the bone and characteristic is osteological evidences actually helped us in taking good sample, right sample. For example, this tooth sample had so much of amblyopy that it had more amblyopy DNA than the human DNA in this part of the tooth, and this is consistent with the low body temperature preference of this bacterium. So this could be directly sequenced without any enrichment. But for rest of the sample, it was important to remove the contaminating human DNA or other bacterial DNA which decomposed the human body after the death of this unfortunate person who died with mm, leprosy at that time. I'm not saying that that person died due to leprosy, but it died with leprosy at that time. So you can see that these characteristic deformities can be traced back to very characteristically to their bones. Uh, the sinking nose and the damage to the nasal spine. So for that, there was this enrichment carried out. I think for exome capture also the similar approach was actually used to remove unwanted intron and the indigenic regions. So by capturing on this type of slide, you can capture the target DNA. So amblyopy library was prepared from skeleton sample, from patient samples, and from different clinical material that were available from different parts of the world. For Europe, since we didn't had any patient sample, we had to use these skeleton samples from the DNA extracted from these bones in clean labs because it's very important to avoid contamination. And these were then captured on a slide which had oligos targeting amblyopy DNA, and then it was sequenced. It's very much similar like the sure select approach for exome capture. The other approach is to remove human DNA, entire human DNA, and whatever is left. So this is the negative enrichment, this is the positive enrichment, but both lead to higher proportion of amblyopy DNA for better coverage of amblyopy genome. And with that, we were able to see that uh, 16 genomes were published in this study. And what we found strikingly was that this ancient DNA sample SK2 from this skeleton sample from UK, it had only 28 differences in a time frame of 1,000 years from the another sample called NHDP 63, which is uh, also present in patient samples as well as in armadillos. So we call it as a V1 genotype or genotic strain from southern United States, and they are exactly identical even after 1,000 years of their divergence. So that further proved that the same strains actually cause disease in these animals and in patients. So what does it mean? Can anyone tell regarding the if anyone would like to unmute and give a conclusion about whether these strains from 10th century Europe were pathogenic or not? Anyone? So that
So it looks like that.
I think they were non-pathogenic. Yeah,
no,
I
they,
believe.
they, they, they basically because they are the same strains which cause disease even today. So if the same strain is causing disease today, in these skeleton samples also they were causing the same type of disease that we is currently present in animals and in humans. So that proves that disease uh, declined not due to the loss of virulence because the same strains are still equally virulent in America, because those strains actually came with human migration through colonization of America by European population, and those strains still perpetuate the same disease that was present in medieval Europe. So basically, that ruled out that the leprosy decline in Europe was not due to the loss of strain virulence in medieval Europe. So some other factors such as host genetic factor or demographic factor or both might have contributed in a more impactful or effective manner. So this was published in journal Science, uh, because this was an active collaboration between archaeologists, molecular biologists, ancient DNA work. So Shunman, Verena Shunman, she is PhD student of uh, she is PhD student from the lab of so she basically did her second PhD actually for this, including this work actually, and this is from Swante Prabhu's legacy actually. His postdoc was the corresponding author for this work along with Professor Stuart Cole. So we had three corresponding authors for this publication and four joint first authors for this, representing different skill set that they brought. So some were ancient DNA biologists, others were um, from the our lab mainly contributed about the biology of the disease and the patient samples, and the skeleton samples were provided by archaeologists separately, and then the bioinformaticians have been making sense of this entire data. So in this 1,000 years of investigation, we found only one species of leprosy bacterium, that's Mycobacterium leprae, and we also saw that it's from Europe that leprosy reached America. But there is a unique form of leprosy in America, which is known as Mycobacterium lepromatosis, uh, and it causes diffuse lepromatous leprosy, which leads to this type of loss of oxygen supply in peripheral parts of the body because of the damage to the blood vessels. And this type of characteristic loss of eye hairs, known as madarosis, that's not unique feature of only leprosy, but it's also present in other forms of disease. So we didn't know why this is so severe form of leprosy, and in one of the sample, it was shown that the leprosy bacterium DNA is very divergent from the existing genomes or strains of M. leprae. So it was later on found to have more than 9 or 10% genetic differences. So it was difficult to believe that this is the same species. That is why it was important to sequence its entire genome. And around the same time, it was reported in squirrels. The declining population of squirrels was being investigated for pox viruses, and some of the squirrels had these characteristic lesions of leprosy and were full of acid fast bacterium or mycobacteria. And from that, it was found that it is another species of Mycobacterium lepromatosis. Uh, So its however its genome was not known. It so entire genome was sequenced for this bacterium. Professor Han had first described it in 2008 from the autopsy sample, because this patient who died of this disease was having large number of acid fast bacilli, but it was not Mycobacterium leprae. So they wanted to know what is this causative agent, and they reported it as a Mycobacterium lepromatosis. And the first genome was published by our group in guidance of Professor Stuart Cole. Currently, Professor Stuart Cole is the president of Institute Pasteur. So by comparative genome sequencing, we were
Sir,
able to.
why you are not working in plants? Why you have chosen this animal pathogen, human pathogen?
Um, so it's I think every field is important, and since my training was in this field, I have been fascinated with this ever since. Actually, I entered this field. I'm so currently we are not only limited to Mycobacterium leprae, but also to viruses and to other pathogens also, such as scrub typhus. So basically, we are on the microbial genomics currently, microbial pathogenesis and genomics. Uh,
And only animal pathogens because you are
Not
in.
necessarily, because I was talking about Verena's work in this paper, and Verena has done this for the ancient seeds also, because just like these skeleton samples, you would also see some from the skeleton remains, and in the excavation, you would also get to see some seeds from 1,000-year old. So what are those seeds? What are their varieties? So this was a main thesis work of Verena, but yes, it requires different skill sets also, and she very successfully utilized her uh, skills in genomics for studying human pathogens also. She studied plague also, and now they are again applying for ancient plant pathogens also. So it's actually so versatile tool actually. So I don't restrict myself to one part. And in recent Bioclues talk, I was actually just talking about the diversity and the magic of four letters. So yes, plants are also made up of the same ATGC actually. So and the same genomic sequencing technologies and molecular epidemiological tools can actually be very much applicable for any plant pathogen or even plants also. And this is the actually the era of one health, and that is the beauty also. So yes, I agree with you that one should be guided by their observations and then research questions rather than limiting to one particular area only. So yeah, coming to the uh, comparison of this pathogen, Mycobacterium leprae and lepromatosis, since this is also not a culturable organism, just like Mycobacterium leprae, we had to get clues from its genome about its biology. For example, there are large number of pseudogenes in both the organisms. The GC content is very similar, uh, and it's lower than other mycobacteria. Whereas in, for example, in other mycobacterium, it's around 65%. In M. leprae, it is around 57%. Interestingly, let's come to the molecular epidemiology of leprosy again. When you see all these ambulatory strains from the science paper here, uh, when you compare all these strains with amblyomatosis genome, you would find them almost identical, clustered here as a single line. When you zoom in, you would see that they have some 5-10 differences in between themselves, among themselves. But with amblyomatosis, it has nearly 300,000 SNPs, 300,000 compared to just 30 SNPs between amblyopathy strains. So if you back calculate how long ago these two species diverged, this comes to around 13.9 million years. So, and as we discussed that anatomically modern humans are how old? Roughly, is it a million year or less than a million year or several million years?
Less than a million years.
Yes, it's only 100,000. It's 0.1 million years old. So we came to on this planet very, very late. All these pathogens have actually been there millions and millions of years before us, and they have persisted in nature. They don't need us actually. They when people say that we would eradicate this disease in such a short time, and there are many programs which are named as for example, National Leprosy Eradication Program. So eradication is a very strong and for this, for the diseases which have genetic reservoir, for example, armadillos and these squirrels have been reported to have this. Subsequently, even some chimpanzees have been also uh, shown to have this disease in nature, in wild. So, for these diseases, WHO doesn't consider them as the eradicable diseases. We need to understand this difference, and we need to understand the diversity and evolution. These have persisted for so many years, 14 million years, and we, until just 10 years ago, we used to consider them as the same organism. But genomics gave us insights about deep divergence of these pathogens. And if you consider it further with M. tuberculosis, it's around 60 million years ago. Imagine the time and the type of life on Earth at that time. So, genomic insights into diseases. So, for example, for this particular pathogen, we don't do genomics for the sake of doing genomics, or just because we have these wonderful machines which can produce high throughput data, but to understand whether this species also have neuropathogenic potential. For example, the same genes which cause nerve injury in mycobacterium, because in M. leprae, or because of the genes due to which M. leprae is able to cause nerve injury in humans. The same genes are present in this Mycobacterium lepromatosis also, with more than 85% identity. So that shows that even if we are not able to culture this organism in any artificial medium, it shows that this is also present. This ability of causing nerve damage is also most likely present in this pathogen. Further, there was some experimental evidence of the presence of this species inside the nerves. So Mycobacterium leprae is one of the has this unique ability to invade nerves. No other pathogen is actually known to, or no other bacterium is known to invade nerves. But this lepromatosis was also found inside the nerves, so that further substantiates the observation from the genomics. Yes, but we have to design the experiments so carefully actually that this laser might capture microdissection, cut out only that particular area where the nerve was having amblyopathy from the inside and not from the outside of the tissue. And from the PCR, we were able to show that this actually causes nerve damage also. And there are no plasmids to account for higher pathogenicity of this organism. So it is probably the host response that leads to higher damage in certain populations. So those are actually certain take-home messages from this work, that that genomics can give us a lot of insights in form of molecular epidemiology, in form of genotyping scheme for tracing the uh, pathogen transmission dynamics. And if you would like to remember 3 or 4 messages from this work, is the natural leprosy of armadillos is a zoonosis in southern United States, which leads to local transmission, and people need to be careful about this type of exposure. Having this in the public domain actually sensitize the doctors also to have this as a clinical suspicion when somebody comes from the area with this type of presentation and has some direct or indirect exposure to armadillos or the areas where these infected animals are present, then doctor would be also uh, suggesting this type of diagnostic tests. Otherwise, it used to take more than 2 years before actual detection of pathogen or suspicion for leprosy could be done. And that leads to transmission and damage to the tissue in person. And sometimes it can also lead to disability. And the disabilities are the main factor for social stigma. People are afraid of leprosy because they see that even after curing, those have certain damage in their hands, and that leads to social stigma. And the next is the current strains of leprosy reached America via colonization. We discussed that India was not the cause of spread of leprosy in the rest of the world, and I don't want to blame any particular country. So we haven't even focused more about tracing to the origin of leprosy to a particular geographic area, which may not be very accurate also. And WHO also came up with this type of recommendation during the COVID time to delink the geographic origin. Earlier, it used to be named as Indian strain, then UK variant, and like that. But they came up with this type of alpha, beta, delta naming system. And it's important to be careful of our terminologies because they have long-term impacts. And then again, this molecular epidemiology should not be used as for the blame game. This is not our tool to point out fingers at others. These are pathogens, they have been there for millions of years. And having simple tools of PCR based sequencing or that can actually help us understand their evolution and uh, can help us guide our behavior also. And disease agent can remain present in wild animals for centuries undetected. For example, these red squirrels, they carried this strain for 700 years and nobody even suspected this actually. And nowadays, probably for some reason, those are not actually contributing to transmission to humans, but it's good practice to keep away from them. Leave them alone. Leave armadillos alone. Leave these wild animals alone. And rest things can be done by taking precautions when coming in contact with these. So yes, I think I was telling that I would keep it very brief, but I still have get gone carried away with all the fascination that I still have with this subject. And I would be happy to take any questions at this stage. After which we would basically take some hands-on the carbon code aligner only for 15 minutes.
I'd like to ask one question. Do you have any other kind of or similar kind of diseases in zoonotic origin of India?
So
Like
for leprosy,
we have cats and dogs at home,
mhm.
and
So
mostly we
mhm,
don't know whether it is a domesticated cat or it is a wild cat
mhm.
because
So do they carry leprosy, or the question is that, or they carry other pathogens? That is the question.
both.
Okay, so yeah, so that is the last message of this take-home message, that disease agents can remain present in wild animals for centuries, undetected. But for the pets, actually, if they are being regularly checked, given some vaccination, that can be a safe practice. Uh, but for wild animals, it's better to leave them alone. We
Like
don't know what they may carry and
no,
how.
actually, some animals we don't purposefully take them.
Mhm,
Like for example, uh, I don't like pets. I don't like to have pets in my family. None of us rear pets. But the thing is, like for the past 2 3 years, group of cats or one or two cats they used to visit my home regularly, and something means the newborns of the first cat settle in my house. So this is happening for the past 2 3 years. During this lockdown corona period, I was alone, so they thought like this is the best place to occupy, so they come and settle in my house. So
mhm,
now also I do have two cats, and at least half of the day they will be in my house only. Night they take occupy one room totally.
mhm,
They won't even allow me inside. So it is like that.
mhm,
So I'm afraid after
mhm.
seeing this presentation. I'm afraid whether I
No,
will.
no, it's not the purpose of that actually. I think if they are starting to live there, and if it is possible to get them vaccinated for certain things, for example, I don't know, maybe rabies, if it is useful to have
No
them
dog.
vaccinated.
It's not dog. It is a cat.
So I basically, if there are some vaccinations available for common pets, and if those are applicable for cats also, then it's good to get that. But
No, it
it's
is
like
a wild cat. How I will be able to carry them to the veterinarian, and it will be very difficult. It
okay,
is wild, but This
If it is,
4 month old,
mm,
two kids are there.
mhm,
They are some
mhm,
5 months old, hardly 5 months old.
mhm.
Um,
No, I think it's okay. I don't think that there is so much concern about because human civilization has
Actually,
actually
it is a newborn.
been
Means
exposed.
since its birthday, it is in my house. Means it was born
Mhm,
in my house only. So since its birth, it is in my house. And both those
mhm,
boys, mother
mhm,
left them
mhm,
with me last month.
mhm.
So for the past 1 month, only those two kittens are with
So
me.
those are actually not then actually wild cats actually because those are like kind of raised in cap like in. So
Yeah,
I don't think it's
they are
so
in
much.
isolation. They don't have any contact with any other animals.
So that
Mostly they are in my foyer, and then
mm,
they go to backyard of my house. So only that much it is.
mm. I think there is not
After
so much concern.
seeing this present, no,
Mm,
sometimes they jump on me and they occupy one full room. They don't allow
mm,
me to enter.
mm.
So that kind of thing.
Since now they are familiar with you, it would be good to ask a veterinarian what type of precautions can be taken in terms of vaccination. They maybe can be probably if they are actually cohabiting. Uh,
I think after growing up for a few more months, they may leave me.
yeah,
But
that is also possible when they have familiarized
now
with the
they are
outside.
small babies. That's why it is with
Mhm.
me.
Yeah,
Yeah.
Yasin, keeping in view of brevity of time, because Pushpendra has to leave. There are a
Yes,
couple
sir.
of
Yes, sir.
questions from
Sorry, sir.
others. Huh?
Sorry,
Thank you.
sir.
Yeah, yeah, yeah.
Mm.
Yeah, Sandeep, quick question, please. I also have a question, so yeah, please quickly. Yeah.
सर एक्चुअली जो माइक्रो आर्मेडिलो में जो लिप्रे है दे आर नॉट कॉजिंग एनी डिसीज टू देम एंड दे आर
No,
लाइंग देयर फॉर वेरी लॉन्ग
no,
टाइम.
no, 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 no. They do cause similar type of disease presentation in armadillos also. They do develop nerve damage. I didn't talk much about that today because I had to cover three different stories. But they do cause disease in armadillos also.
ओके सर दैट्स ऑल.
Yes, it's pathogenic to them.
Yeah. Yeah. Next one, please. Yeah. Yeah, Deepika. Yeah.
Deepika, yeah. Sir, I want to ask that, like for pneumococcal disease, we have developed vaccines which are given to the baby. So, there is no vaccine that which can be given to the babies so that they do not develop this leprosy disease.
Um, in fact, there is no very proven vaccine um, for leprosy. It's BCG provides some protection against leprosy also, cross protection. Uh, but in recent years, there has been one Micrococcal indicus pranae as a vaccine, but it is more for a therapeutic purposes to as an immunomodulator. So currently, there is no vaccine under the program for leprosy, but BCG gives some protection in early stages, just like in tuberculosis. But yes, at present there is only immunotherapeutic vaccine called Mycobacterium indicus pranae in the honor of Professor Talwar, GP Talwar from NII. Yes, any next question, please? I hope I have answered to some extent.
Good evening, sir. I have one
Yeah. Thank you
question.
so much,
Mhm.
Pushpendra. Harsh, do you have a quick question, please? Yeah.
Just a quick question, sir. Sir, you said that we cannot culture this leprosy bacilli. So why is it like that? Is there any
Uh-huh.
limitation? We don't have
Uh-huh.
any kind of media for that.
Yes, it's actually this bacterium has got so many pseudogenes that it has become host dependent, or it started living in. So earlier it might have lived in some free living organism like a soil bacterium, but after reaching some highly specialized host environment, for example inside the nerves where it has no competition and everything is being supplied through host, then uh, the principle of use it or lose it might have led to this uh, pseudogenization that has. So more than half of the ambulatory genome is actually having either pseudogenes or intergenic region. So probably because of that, a lot of its metabolic potential has been actually compromised. So that might be the major reason of its slow growth rate and failure to cultivate it. But genomics can help us in a lot of understanding of its biology.
Thank you, sir.
Yeah, I'll have a quick question, Pushpendra. I'm sorry. That
Mm.
was a wonderful talk. Always a great learning experience. Whenever you speak, you start right from the basics. That flow is absolutely amazing.
Thank
In
you.
fact, um, Yasin, she stole my question. In
Mhm.
fact, what I believe is, I mean, you may kindly correct me. The dogs and probably the cats, they share something what we call it as oxytocins, which
Mhm.
match with our eyes. This is a kind of a chemical, that is probably stimulated by their eyes, and so as our eyes, and that is probably why we call dogs as best friends. Why don't we breed snakes, or why don't we probably breed
Mhm.
tigers? Because they don't emit
Mhm.
that kind of oxytocins. But there is a
Mhm.
missing link between
Okay.
the
I
oxytocin
didn't know that.
stimulation and the horizontal gene transfer of events, where some of these particular genes were probably resistant to certain diseases like leprosy. So during your armadillo's study, is there any kind of missing link or cross connection between these resistant genes that are probably evolutionarily transferred, say from bacteria to the host or the other way around over the years?
There is only one example of
Yeah, Pushpendra,
gene.
was I clear? I'm sorry. Yeah.
Yeah, there was some interruption, but whatever I could understand, I am trying to reply. I'm telling that during the co-evolution with eukaryotes, there are certain genes which have eukaryotic origin in M. leprae. And in the other mycobacteria, they have prokaryotic origin. For example, Pro-S gene is one such a gene. And besides that, there are not many examples of horizontal gene transfer between human and leprae. And that is probably because with humans, this bacteria has evolved for a very small period of evolutionary journey, considering its evolution around 20 million years ago. Uh, and since then, it has changed very little only. It has not changed much. I think your video is frozen, Prash. Am I audible?
Yes, sir,
Yeah,
you are
yeah, yeah,
audible.
very much. Yeah.
Okay.
How about now? Yeah,
Yeah, it is
yeah,
now.
yeah, yeah, very
Okay.
much, Pushpan. Yeah,
Okay. So basically,
yeah,
it
yeah.
has
Thank
some
you, thank you.
horizontal evolution, horizontal gene
Yeah,
transfer,
thank
but
you so much, Pushpan, for the.
okay. Yeah. Yeah. Okay. So any other questions or?
No, that's it, Pushpan. Thank you so much. If you have time, you can quickly demonstrate for the next 5 minutes. Otherwise, I think, I know you and your lab are in a celebratory mode. That's a big day. Heartiest congratulations, to you and your, lab members. 4 years is an excitement, I would say. And I usually, keep giving your NEGM paper, to our Amrita University students, actually, to read. That's a wonderful paper, the way, that it was carved out, because, we have a lot of, microbiology and biotechnology students as a part of, research methodology, the PhD courses that I teach. And even for, basic level BSc and MSc students, I give them, this particular paper of yours. Although I'm not a microbiologist, but, I find, immense pride in reading that particular paper. Thank you so much, for that great presentation and papers all through. Yeah. So friends, I think Pushpendra has a big day, today and tomorrow. Says he'll be going there, but he'll probably try to give a quick demo on the tool. Pushpendra, do you think you will give a demo, or we can do it maybe at a later
Uh,
point of time as well if you're busy?
Maybe
Yeah.
just a 5 minute overview of what the tool is actually. It's not a fully
Sure, sure.
free tool. It's actually for 1 month you can use it, and there are many other alternates also, some of which are free. But since I have been actually very much familiar with this, and I have actually found it very user friendly, so I'm just introducing it to you. During my postdoc work also, I got this license awarded from this company. But for 1 month you can always use this on one computer, and if you need to extend it, you can install it on another computer also for another 1 month, like that. So this code on code aligner actually, and for all those studies we needed small alignments of PCR sequences, PCR after PCR sequencing. So basically, in after you have installed this, it would actually ask as a open as a new project file, new project. You can say as a new project here, and if you want to save the previous one, you can save it with whatever name you want. Let's say test one Bioclues, but and then it's actually opening as a new. So this would be the new project that you open in, and this is for the Sanger sequencing uh, based alignment. It might be very preliminary for you, but I think all the studies that I have explained to you, more than 90% of the strains were actually compared by using this approach for identifying which SNPs are present in armadillo samples, which are present in skeleton samples. Not for skeleton samples, because there we only used whole genome sequencing approach, but for most of the clinical samples we used PCR sequencing of target genes. So when you have any Sanger sequencing results, you can add either as a sample here in a new project. For example, in this folder, I have saved certain results for this workshop, and those are the raw files that you get from the Sanger sequencing run.
So excuse me, sir.
You
I
can
think
select them all.
so. Your window is
Okay,
not visible. I think
okay. I'm sorry. I didn't. So
your
far, you haven't
your
missed
screen is
anything.
visible, but the window which you are working on is not visible.
Okay. Is it because it's a window inside window? So it's you are probably. Let me see if I can. Okay, so I'm stopping the share, and um, maybe I would just point one window. So window one, maybe it's this one, quote unquote. Okay. So is this now visible? It's starting to visible to become visible now.
Yes, sir. Yes, sir. Yeah,
Yes.
it's visible
Thank you
now.
for pointing that out. I didn't start anything on the program so far. So this is the window that you would open, and in the file menu there are different. You can get the tutorial also for the next presentation on its website, and it has also some uh, examples files which you can explore by yourself. I'm just familiarizing various versions to help you move with that tutorial. So you can open up a new project, and that would look like just like this. Um, uh, so you can open, since there are no samples, you can import entire folder on this screen, import folder. So if all your samples are in one folder, you can simply import it. For example, in the downloads, I have saved this folder. So do you want to import all 103 files? Uh, and you can say that do not show me this type of simple warning. So it's now importing all 103 files. There is a Word document that would automatically be not imported because it is not in the desirable form. So out of 103, there was one Word file which it didn't import. Remaining 102 some files are imported in this folder. And now you can see what are their contents, their names, their length, their qualities values as per the Fred score, and their you can click one by one and see how their chromatogram looks like in Sanger sequencing. you can see that there is some poor quality in the beginning, and in the ends also you would have this type of poor quality of sequences. By you will see that all these are actually poor quality sequences here, which need to be trimmed. You can do it one by one for all these samples, and sometimes you may not even know whether this is of particular gene, or if their mislabeling is also done, then also you would not know which sample to assemble together. So if we want to perform it quickly, to we can simply uh, use this feature of assemble. Just click on this symbol assemble, and it's now assembling the sequences based on their similarity. The same genes would be assembled together. For example, in this config number 1, it has automatically grouped all the samples which have similar sequences. For example, these are the measles virus samples from Jabalpur. Recently, we sequenced these samples from the measles outbreak. And in that, you can see that there is this alignment. The green highlighted area indicates that the sequence quality on the ends is poor, and whenever there is some snip, it is indicated, or some mutation, it is indicated in a different color. For example, this T is different than the remaining two C here. Are you able to see the cursor? I don't think you are able to follow the
No,
cursor.
sir.
No, sir, it's
Sir,
not visible.
actually,
Okay,
you are
so I
visible.
think, okay, I need to, I think, again stop presenting and share the entire screen. Anyway, let's entire screen. Let's see if it works. But with the entire screen, okay. Now I think now you would be able to see the screen here. This one. On this screen now, are you able to see the cursor?
Yeah, sir.
Okay. So wherever there are snips, you would see them in different color of base. For example, here it is C. The chromatogram is also visible. That blue color peak is indicating a cytosine C, and just below it there are different strains which have a T, and whether it is true or not, you can see that the T is very clean here. It is truly a uh, different base here, and most of these have a clean base as a SNP. Um, so like that, you would be basically able to perform the sequence alignment, and you would be able to identify the SNPs. Once you have put it into a right frame by the help of some reference sequence, you can convert it into bases also. So now it has converted into amino acid sequence, 3-letter amino acid sequence. So this is serine. So let's imagine that without wasting time, I'm not going into the frame correction right now. Once you have, based on the reference sequence, you have identified that this is the correct frame of translation. If it is not the correct frame, you can convert it again by clicking on this area. So now it is amino acid sequence that you are able to see. And if it is not the correct frame, you can select the next frame here. So now it has selected next frame, next frame, next frame, and then it would be on the reverse strand by reverse complement, and the reverse complement can be done here. So everything has now changed into reverse complement, and you can choose the frames here by clicking on the next frame. Once you are sure that everything is perfectly aligned and there are no insertions and deletions, for example, this type of, we are not talking about these type of gaps. Let's focus on the clean area where there are no gaps. And then you can see that this serine, there is a change from leucine to serine. And what is the base change? You can again come to the base uh, on here. Is cursor visible? I think cursor is visible to you.
Yes, sir. Yes.
Okay, so on this base translation, you can see that it is TCA that has converted into TTA, and you can again change base to, so that leads to the change of leucine to serine. And if you are sure that everything is correct and you don't want all these bases inside your alignment, you can just select them and just press delete on your keyboard, and that deletes all those bases. And you can even make phylogenetic tree here, build tree. If you are satisfied with everything, you can select which option, whether neighbor joining, or if you go by the default method and you want a branch, label the branches also, you can select that and build tree. So now you have this phylogenetic tree of these sequences. And what good thing is that it has automatically reverse transcribed some of your samples, because this was from the reverse primer. It has automatically made a reverse complement of the sequences which are shown in the red color. Are you able to see the cursor? This here, the sequence is in red color. Prash, can you reply? So yes, this is
Yes,
basically
yes, sir.
okay.
We are able to see.
So yeah,
Yeah,
so
yes,
this is
please.
very,
Yes, please. Yeah.
so this I find it very useful, so that I don't have to manually convert the sequences into reverse complement, which is also error prone. So yeah, this can be very helpful for identifying the SNP and its effect, whether it is synonymous SNP, on which position it is. Once you know the reference position, you can identify. So based on this SNP identification method, we put that into the phylogenetic tree for which was used for the molecular epidemiology. And you can compare any sequences of plant sequences or virus sequences or bacterial sequences. For example, the other country has the rubella virus. uh, if you want to trim this for the low quality basis, you can add it, and uh, in the file you can choose to trim the sequences in this folder, and you can rename this is rubella virus, so it would be easier later on. So now inside this, if you want to trim the sequence, you can choose in the edit section. So there are several ways to remove low quality bases here. For example, in the unassembled bases, you can, there are still only just two samples for, yeah, there is this option called clip ends or trim vector. So these are already trimmed, that is why this option is currently not visible. Clip ends means low quality bases can be removed. You can unassemble this sample also to put it back in this unassembled, and there you can trim, you can clip ends. This is the option of clipping the ends. So with this option, it will indicate that how many bases will be, how many samples will be clipped, what would be the average length before clipping, and what would be the average length after clipping, and how many samples will go to trash after this clipping or uh, removing the basis of low quality. So just then accept clip, and that after that you would have a better alignment, because low quality bases or unreliable bases have been automatically removed from the ends. And later on you can also see the alignment, and inside this config, when you see that that's first converted into nucleic acid, and you want to see that whether this G is actually a SNP or not. So it would open in the chromatogram, and you will see that this is not actually a very good SNP, good base, because here there are more than one peak. So you can put that as a N. So now it is not a very high quality SNP. So you can ignore it and call it undetermined SNP, undetermined base. But if there are some, for example, this is a very clean area, all others are T and this is only C. So let's click on this, and this is very clean chromatogram of C. Is this visible? Uh, so basically, you are able to confirm that whatever you are calling is true or not, and whether this is actually a tree or not. This is not a very good position actually to reliably say that this is an insertion of T, because the chromatogram is not clean. So you can directly delete it. And similarly, like this is not a very clean signal of T here, because the base is very small. So you can trim it from the consensus sequence like that, and you can basically um, interpret the results well. And only after this type of correction, you should be doing the phylogenetic tree analysis. Okay, so this is all from me from for today. I'm stopping.
Sir, can you show me from where you have downloaded that folder which you have uploaded here?
No, this is from our lab actually. I can share you some of the sequences on the in one of the folder, and you can and with the code on code tutorial there comes some sequences which you can practice from there also. And if you have any question, I can send you some sequences for practice.
Yes, sir. In installation files, there are few
Yes,
example.
yes, exactly. So that would be also good to use those. Any other
Means
question?
we cannot get the sequence from means general publicly available domains like NCBI or
Sometimes
something.
they only provide text sequences. Sometimes they only provide the text sequences, not the chromatogram files. If they are providing chromatogram files, yes, you can, and I can share you some sequences for practice. Uh, so if there are no availability of the chromatogram files, uh, because the text sequences would not be actually very helpful, because you cannot see the chromatogram, or the peaks cannot be seen in the text. Any other thing or question?
Sir, means I have one question.
Yeah.
So when in this experiment, when they are claiming that this DNA is say 1 million year ago or 10 million year ago, so what are the basis for that? Are they
No, no,
using?
it didn't say that this DNA is 10 million years ago. It
I mean the
it
species
it
evolution or
says
something.
that these two DNA uh, are evolutionarily apart for 13.9 million years. These two strains of Mycobacterium leprae and Mycobacterium lepromatosis have so many SNPs that if we go by the SNP frequency of M. leprae, uh, we can backtrack what was their common ancestor, when it might have been there in evolutionary time frame. So that would be around 13.9 million years by going by the SNP frequency or mutation frequency of M. leprae.
Yes, that what same I am asking, sir. That whether they are using any power equation or any algorithm for that to
It's
estimate
actually based
frequency.
on the
This one.
timeline. We know that this sample was taken in 1980s. This sample was taken in year 2000. So there is a 20-year time difference between these two samples, and they are of the same genotype, and they have acquired, let's say, 25 SNPs. 25 SNPs in this much time period of 20 years. Like that, when you perform it for the whole genome data, it gives you an estimate, uh, posterior probability uh, of how long ago they might have had a common ancestor. Because we believe, so it is based on the assumption that all sequences emerged from some common ancestor at some point, some time point. When that common ancestor might have been, that we back calculate based on the SNP frequency.
Okay, sir. Thank you, sir.
Okay. Yes,
So I think we should stop here and thank Pushpendra for his gracious time and presence.
thank you very much.
It's
I
always
really
nice
enjoyed.
hearing him.